Hello everyone, welcome to chemistryATLAS. This is the first video in the channel. Before continuing with the video, I would like to say that in all of the upcoming videos, we'll be covering most of the topics related to the CSIR and GATE syllabus. So if you are new to the channel, feel free to subscribe it and hit the bell icon. In each of the videos, our main motive will be the discussion of the important points related to the respective topic, which will help us to analyze and solve various questions. We'll also be taking the previous year questions in the later videos. So let's start with our today's topic, hemoglobin. Speaking of hemoglobin, it is an oxygen transport carrier. That is, it helps in the transport of oxygen from the lungs to the different parts of the body. That is, it acts as an oxygen transport carrier. It is a large intracellular protein which is responsible for the red color of red blood cells. This hemoglobin consists of a porphyrin ring, which is a heterocyclic ring system containing 4 pyrrole groups joined by methane bridges. When we will draw the structure of hemoglobin, it will be cleared what are the pyrrole groups present there and the methane bridge which we are talking about. Next, each hemoglobin molecule comprises of 4 heme units which are bound to a protein chain on its surface. Now, what is this heme unit? So if you draw the heme unit, this heme unit is generally the heme B unit. This heme B unit, if you draw the structure of the heme B unit, nitrogen, 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 and there will be the ferrous ion coordinated to the 4 pyrrole groups. These are the 4 pyrrole groups, and these are the methane bridges. These are the 4 methane bridges that are being coordinated. And there are substituents present is methyl group. Here we have the ethene group. Here we have again the ethene group. methyl group. Here we have CH2, CH2COOH. Here we have the CH2, CH2COOH. There is one methyl group. There is one methyl group. So this is the Hem B unit. So like the Hem B units, there are other Hem A as well as Hem C, and they are generally differentiated on the basis of these groups. Depending on the substituents present in these positions, they are categorized as Hem A, Hem B and Hem C groups. So now when you talk about the different forms of hemoglobin, we have various forms like the deoxyform, oxyform, then hematine, then hemoglobin. So what is the deoxyform? The deoxyform and the Hem B, there is one nitrogen. It is attached to the nitrogen atom of imidazole cyclone. Okay, in the deoxyform, this is generally the deoxyform where there is no in which the iron center is not coordinated to any oxygen atoms. In the oxyform, the iron atom coordinates to the oxygen in a band fashion, and this angle is found to be 130 degree. And in the later videos we'll be talking about the hematine and methemoglobin. So for now this is not required. So if we talk about the different important points of the deoxy and oxy forms, we have in the deoxy form, iron is in +2 high spin oxidation state, and if we talk about the oxyform, here iron is present in +3 low spin oxidation state. And in the deoxy form, this ferrous ion is slightly present above the plane comprising the 4 pyrrole groups. As a result, it leads to a square pyramidal shape. But in the oxyform, this iron, the ferrous ion gets converted to Fe3 plus, as a result of which, it being smaller in size, it fits well into the cavity comprising the 4 pyrrole groups, that is Fe3 plus. So it leads to a square planar geometry. Next, in the deoxy form, it is purple blue in color. The deoxyhemoglobin is purple blue in color, and the oxyhemoglobin is bright red in color. Okay, then there are two states of hemoglobin. Two states of hemoglobin. Number one is the tense state or the T state, and number two is the relaxed state or the R state. This T state has less binding affinity to oxygen molecule. So in other words, we can say that this tense state refers to the deoxy form, and the relaxed state has high binding affinity to oxygen and so represents the oxy form. Moreover, it is important to note that since in hemoglobin there are 4 heme B units, 4 heme B units, and as we have seen that in one heme B unit there are 2 carboxylic acid groups. So in presence of a base, this heme B unit will act as the source of 2 acidic protons. So overall, if we talk about one hemoglobin molecule which comprises of 4 heme units, so overall we'll get 8 acidic protons. 8 acidic protons. Okay, 8 acidic protons. Now the 4 heme B units has a tendency to accept one oxygen molecule successively. So after the first binding of the oxygen of hemoglobin, the capacity of the hemoglobin to bind with the second oxygen increases. Consequently, the third and fourth binding of oxygen to hemoglobin increases further. This is known as the cooperativity effect. Cooperativity effect. That is, if we denote the binding capacity of oxygen to hemoglobin in the first case by K1, K2 in the second case, K3 in the third case, and K4 in the last case, that is in the fourth case, then K1 will be less than K2, less than K3, less than K4. Okay. Now, talking about hemoglobin, which comprises of 4 heme B units wrapped around by protein chains. These protein chains may be of two types, that is alpha protein chain and beta protein chain. Alpha protein chain and beta protein chain. Now, if the sixth position of the beta protein chain where glutamic acid is present, and if this glutamic acid is replaced by valine group, then we get a mutant hemoglobin. Which is known as hemoglobin S. This hemoglobin S is responsible for the disease sickle cell anemia. And one more important point which I have forgot to tell you in case of this the different states of hemoglobin is that in conversion of the 10 state to R state, which is facilitated when 60 water molecules bind with deoxyhemoglobin. So this is all for the today's video. I hope you like this video, and in the next video we'll be dealing with the other forms of hemoglobin. Thank you.